Okay, this is where we left off previously, and we're looking at neural mechanisms in terms of proximate level behavior. So animals have a nervous system of one form or another, unless you're a sponge, and so the nervous system, whether it's a neural net or a central nervous system, like invertebrates, it's obviously involved in making or producing behavior. This is kind of an interesting case. There's these larval blister beetles, as we see here, that are all around here. And what they do is they mimic a bee's female's pheromones, just like that orchid did. In this case, the males come along, I mean the bees come along. Oh, it's actually a male bees, yeah, system. So they release a bouquet from the female, a mimic of that. And then the male comes in. He's getting infested with these little larval blister beetles. So in other words, the nervous system has kind of been hijacked. The larvae crawl onto the male. He then finds and mates with a real female. Unfortunately, they crawl onto her. She flies off, transports all these blister beetle larvae back to her nest, where they eat all of her offspring's provisions in their brood chambers. Okay, so it's a pretty diabolical sort of system in terms of if you're a bee, a female bee. Now, I want to get into a little bit more detail with this evolutionary arms race that's going on between moths and bats, and different kinds of moths and different kinds of bats. There's been this arms race that's going on between bats and moths. Obviously, it's an antagonistic relationship. The bats are trying to eat the moths are trying not to get eaten. The bats use echolocation, which is a high-frequency ultrasound signal that we can't hear to be above the audible for us. Obviously, other animals use it, like toothed whales. In fact, even baleen whales, I think, use it as well. And not all moths, now some of the moths actually have ears that we've discussed earlier along their thorax, so they can pick up ultrasonic signals from the bats. Not all moths can do this. You almost wonder, well, if they're getting eaten by bats, how come this group evolved it and this other group didn't? Because it's evolved a number of different times independently. If you actually look at a moth phylogeny and which one has ears. The moths also have structures to produce ultrasound, some of them. In fact, it's been discovered that moths actually communicate amongst themselves with ultrasound, just as bats do amongst themselves with ultrasound. Okay? With echolocation. So it's kind of like the electric fields that we talked about earlier with weakly electric fish and strongly electric eels. They use echolocation much for the same reason, to communicate, to use it to find location, especially if you're the fish in a very murky area. if you're a bat in the evening when visuals aren't going to do you a damn bit of good, especially caves as well. There's even some birds that actually use it, oil birds that are in caves, so for the same reason. Now, bats also, as part of this evolutionary arms race, they're competing amongst themselves for food. So oftentimes they all roost together. They all come out at night. They're feeding like crazy on the moths. And it turns out that the bats can actually jam each other's signals so that it gets the bat that's getting its signals jammed is discombobulated and he loses track of where the moths are. Okay? Stealth bats, there's a kind called barbastel, actually, as part of the counter strategy, well, let me get to the moths first. So, they have ears to detect, and they avoid or deter bats from near and far away by a number of different things. Well, they've got these ears that they have, they're tympanic membranes. As we'll see, they're attached to two different kinds of neurons. The ones that moths can hear bats from about 30 meters away. Usually bats can hear or detect moths from further away, but when a bat is far away, a moth will usually just kind of take some evasive maneuver and take off to escape. Now, the thing is, as we'll see with the moths, if a bat is really close, and they can obviously detect that, then there's this evasive maneuver that they use. It's actually a fixed action pattern, where well, we'll get into the exact mechanism in just a second. So, back to the bats. Now, stealth bats, some bats have, obviously, if you can be detected by your prey, then it would, some individuals who could make a softer ultrasound, it may not carry as well, but they've got better structures for receiving it back. If they can actually produce a low-level echolocation ultrasound that the moths can't hear, but they can, then they'll have an advantage in terms of getting close to a moth. So there's this stealth bat called a batastrel bat that does that. And then there's some moths that actually jam the signals of bats. So I'll show you a video of that where a bat is approaching, and right at the point where it's just about going to grab the moth emits an ultrasound that kind of confuses the bat. It no longer can figure out where exactly the moth is. And then there's another of all this kind of things that are going on too. There's some moths that are poisonous, and we've talked about aposematic coloration. Everybody remember what that is, or people who don't? So what's an example of aposematic coloration? Frogs, anybody?
Poison dart frog.
Poison dart frog.
That
right.
alerts that you should eat, don't get near because
right. Bees and wasps, basically, you're advertising that you're dangerous or poisonous or venomous. Okay, well, there's these tiger moths that emit an aposematic ultrasound to bats that's very loud. The bats obviously hear it, and they avoid it because they know that this species is actually toxic. And then, based on this, there's both Batesian mimics of this tiger moth and Mullerian mimics. So a Batesian mimic is a another moth that's not toxic, but emits the same signal as a toxic one. And a Malarian is another toxic one, and so the more toxic ones, models there are out there, the better the deterrence. And then moths obviously mimic bat ultrasound to deceive the bats. So this is kind of what's going on here. In other words, like any good evolutionary arms race, or think about arms races among humans during the Cold War, or any war for that matter. Humans have designed offensive weapons, defensive weapons, to counter offensive weapons, and then there's this arms race. Better technology results in better weapons, both offensive and defensive. Well, the same thing is going on in evolutionary time. Now this relationship is like, 60 million years old. It's a very, very old relationship that's been going on between bats and moths. So they've had a long time to actually perfect it, let's say. And this is exactly what you would expect. You expect a defense, counter defenses, different kinds of defenses, all that kind of stuff. It's just that in this case, the signals are echolocation, or ultrasound signals, instead of, let's say, a visual signal. So the bats emit an extremely loud ultrasonic call that we can't hear. I think we can hear, if we're young and have good hearing, up to perhaps 20 kilohertz. And these signals are sometimes up to 100 kilohertz, something like that. They use the echolocation because it actually creates a picture for them. Sound goes around different objects, and it bounces off, and it gives a bat a very good idea of where things are. They've got two ears, right? So they actually, much like stereo vision, they're able to situate kind of a picture of where different things are. And not just the picture of it, but also texture of it. Obviously how big, whether it's a leaf or a tree or a moth, anything like that. Okay, so they use it to avoid obstacles, to locate prey, and to talk to each other. Now moths have actually developed, some of them have developed what's called an escape behavior. So they've got ears that they can detect the signal from, and then they can fly away with this distance, or they take a more dramatic if the source is near. Okay, so this is, in fact, let's okay, so here's the bat emitting, and it homes in on the moth. The moth might take evasive action. If it doesn't, obviously it's a dead moth. So noctuid moths, we've talked about these guys earlier. It's got a brain. It's got thoracic ganglia, kind of like a central nervous system. It's got ears with auditory receptors. And these ears are along the thorax on either side. And it's this is kind of what it looks like. We've got an air sac, air sac, air sac, air sac. And this is the tympanum, which is a membrane that'll be sensitive, just like our eardrums are, to ultrasound. And then there's these neurons, A1 and A2 receptor cells, that are attached to these. So when there's the tympanum moves and these air sacs are changed in configuration, the moths can actually detect ultrasound coming into them. Now, it turns out that these A1 or A2 there are cells like that in different lineages of moths that have developed ultrasound independently. So not all have. Okay? And then in terms of neurons and their operation, you guys have had, obviously, you've had some of this earlier in 106 and 107 perhaps, and then later on. So we've got the cell body. This is the axon in terms of the direction of action potential. And then axonal terminals, the axon, this is the dendritic. And so this is where you would actually get the synapse, right? So now, here we are with a thoracic ganglia again. This is the sensory neuron that's attached close to the ear. These are interneurons. And then there's motor neurons that they're attached to. In other words, when a bat is close and the moth detects it, it sends a signal, doesn't even go to the brain. There's another signal that goes to the brain. But this is not one of those things you want to think about. And it immediately sends a signal through the interneuron to the motor neuron. And then the motor neuron controls the wings. And one of the methods that the moths use to avoid bats is that when that signal goes through, and it's strong enough, it will it causes the wings to not function properly. Like someone cut off the signal, and it results in the moths taking, well, you would say kind of a controlled evasive maneuver by making themselves kind of unresponsive. And so they start to tumble, and in this way they might actually manage to deceive a bat or escape it. Okay. And these are the auditory receptors. There's two of them. There's A1 and A2. And you can actually look at, here's the A1 receptor, A2 receptor. This is a low-intensity stimulus. Same here. Neural activity, moderate intensity stimulus, and then high-intensity stimulus. Okay? So you can actually see what happens. Now, a lot of times these ultrasounds come in pulses, as you kind of heard with the bats making those chirps. Those are actually a series of pulses that are very tightly packed together. Okay? And so this is then, these guys actually send out signals to the interneurons, and then depending on how strong this actual signal is. The A2 receptor, there still hasn't exactly worked out everything. It seems like the A1 is the most important, but the A2 obviously is just having some impact on receiving signals and then behaving on them. So it turns out that when a bat is still far away, like I said, let's say up to 30 meters away, the signal is less intensive. A1 receptors can still generate action potential, but not strong enough so that it discombobulates the flying muscles. Instead, the moth flies off. When the bat gets very close, the intensity of the bat signals are very strong, and it even causes the A2 cells to start firing. It seems that when A2 cells fire and these messages reach the brain in turn may shut down the central steering mechanism. Actually, it bypasses the brain. So there is brain interaction, but the main thing is when they're close, it requires, much like a FAT, instantaneous action. So it's really important. And it causes very erratic, unpredictable flight by the moth, and in the same way, the bat might not be able to follow it. So here we have, and then in terms of the frequency, you can see where peak intensity is. So low intensities, the peak response is usually 40, about 40, 45, 42 kilohertz. And then high intensity, the frequency is usually between 70 and 80 kilohertz. So, and then you can actually see exactly what's kind of, going on here. In terms of the bat coming and then the moth taking evasive action, but we'll actually look at this in a second. Now, let's see here. Okay, this is a figure looking at a barbicel bat and another kind of bat. So B. barbastellius, and then N. leslieri. So there's two different kinds of bats. Now the barbicel is the one that actually uses stealth technology, just like we have stealth fighters and stealth bombers and all that, so that they have very low sonic signatures, and so it's difficult for other fighters to lock onto them. Well, the same thing is going on here. And so you can actually, in terms of the space that we're talking about, let's see. So this is detection distance, and then this is dB, in other words, decibels. And obviously, in terms of low decibels, the bat can actually hear farther than the moth can, and vice versa. So here's kind of parity. Now, the thing is, this is barbicel, and this is the other bat that doesn't use stealth technology. And if you actually look at, let's see, where is it? Well, I'll use a pointer. Okay, so the yellow is where the bat's picking things up, and then the blue is where the moth is. So if you actually look at, and this is the A1, A2 receptor again, same system. What's happening is, for the barbicel, they can emit these frequencies, and they're much quieter, as it turns out, than this bat, and so they can get much closer. So we can actually see, in terms of source signal, what they're, as far as dB level, the barbicel can get emit these quieter things, but still get a signal of where the moth is, versus this one, where it can get a signal. It needs to be up to like 130 decibels. In other words, that would actually let the moth know that it's being targeted. Whereas this guy is coming in, and the moth doesn't know it's being targeted until the bitter end. Okay, just like good stealth technology does. And this is the same sort of thing. If we actually look, this is the one that doesn't use stealth, and this one does. And here we have distance, right? And you can actually see that the moth can actually track this guy from very far out, let's say 30, 35 meters. But this is the one with stealth technology, and they can only track it out to about 4 meters. Okay? And then you can actually see right here, in terms of the DA1, DA2, these are the A1, A2 receptors. This is barbicel, and this is detection distances in meters. And so the moths can only detect you know, between 4 meters and less, and then this guy from far away. Okay. Now, so that's the stealth technology. There's also, there are some moths, as I mentioned, this is actually aposematic echolocation. These are different species of tiger moths. They have these sound-producing structures. They're called tymbals, and these are different species. This one is toxic. This one isn't. I think this one is to a certain amount. And then you can actually see what the different signals look like. Now, this one is actually going to be using Batesian mimicry. I think this one's going to be using Müllerian mimicry, as we'll see. Okay, there's kind of a lot going on here. So this is a Müllerian mimicry graph. Cytomedia epilastis is also toxic. And then this is Cyana tenera, which is toxic. The black lines are 2 different kinds of bats, E. fuscus and L. borealis. Okay, and the solid line graphs the percentage of tiger moths captured. So now what's happening is the bats are actually aware of what kind of moth is emitting the signal. And once they realize that, they quickly learn that, in fact, these guys are toxic, and if they grab it, they usually drop it. They don't try to eat it. Most of these moths have a cardioglycoside that's the actual toxic ingredient, which is affects the heart, kind of like digitalis. Anybody know what digitalis is? If you're going into medicine, it's one of the oldest beta blockers in terms of making the heart beat normally instead of having extra beats. Okay, so it's actually used for, you know, heart disease. Okay, so and then this is a moth, in fact, that's not toxic at all. Okay, and you can see the number of moths captured up here by these bats is very high, but here, even though they might target them, but then they break off. They don't want any part of them because it's too toxic. Okay, and then let's see, the vertical bars are when they switch to different moths. So they start out with a very toxic moth called Cyana tenera. This one is also toxic, and this is where the break point is. And then the tymbals that are on this one, they actually do it where they get rid of the tymbals so the moths can't hear, or the bats actually. Let's see, yeah, the bats can't hear the moths signaling, and so the bats then go and actually up here and capture a lot of them, not realizing that they're a toxic moth. So this is classic Müllerian mimicry, right? Where you have you have the main model and the secondary model. Actually, they're both models, yeah.
So between night 6 and 8, is that because their signal is different, so they attack them and then they relearn?
Yeah, so they're actually looking, this is for night 6, they've now switched from C. tenaria to this one. So they're kind of relearning, but even though this one gives a different signal, so you can actually see the signal here, right? For C. tenaria, well, this is the signal for this one, okay? And it's different, but the bats are still picking up that, in fact, it tells it that it's actually a toxic moth. And then you can actually see significant differences. So at day 5 compared to day 1, there's a much bigger difference. And then day 11 compared to day 10, there's a significant difference as well. And this is actually a Batesian mimicry comparison. Let's see here. Okay. Now, C. tenaria is the one that's toxic. This one is not toxic, but it emits the same sort of signal that, or at least an aposematic signal saying, oh, I'm toxic too, even though it's not. Okay? And then, let's see, we can... So these are percent moths captured. And then eventually what happens is it looks like the bats actually learn at a certain point that, in fact, these guys aren't necessarily as toxic as they thought. So they learn over time. In other words, there is this ongoing arms race in terms of the bats are learning what's going on. They become aware of the deception that's actually going on here. And then here we have the same thing with the tymbals for the non-toxic one ablated. That means they've been destroyed. And so then the capture rate goes much higher. Okay, and let's see here. Okay, well anyway, you get the idea. So this is actually what's going on. This is tenaria. This is the normal, let's see, I'm not sure. I'll have to look at that. Anyway, but you get the idea. So they're using the same echolocation signal as a toxic moth. And then the bats actually eventually learn. And obviously it depends on, this only works depending on the amount of actual toxic models versus the amount of mimics. So obviously if there's a lot of mimics out there that aren't toxic, whether it's visual signal or in this case the sound signal, it won't work as well. You always have to have many more actual toxic models compared to non-toxic models for this to actually work. It's kind of like the monarch butterfly with the that's what's going on. Okay. Okay. Now, oh, good. So Daniel Kish, he was born with retinoblastoma, which is an inherited cancer of the eye. And he had the first eye taken out when he was like, 1 or something, the second eye at 1.5. They both ended up, it's very high incidence where you actually get cancer of the eye. So they took him out. And so he was blind. Well, it turns out that he kind of remembers this, when he was a kid, he somehow was able to realize whether the sounds he made, that he actually taught himself using these click languages to use that to use echolocation in the same way that bats do. And so as he grew up, he got better and better and better at it to the point where he can actually walk around, ride on a bicycle. He can, if you put him in a place and you ask him, well, what's in front of you? He might say, oh, there's a tree in front of me. And it's pretty much all round all the way up until this entrance, then there's branches coming off. He'll go up to the tree and say, well, this is what I see here, right? He also, of course, in certain senses, he's got an advantage, because at night, everybody else is blind, but he is not. So he can go out, and he loves to do this, apparently. He likes to go out into the woods at night, because he can see fine, whereas everybody else is stumbling around with a headlamp or something like that. Okay? Now, there's kind of a, this is kind of a little background information, but in the blind community, most blind community organizations are giving blind people the skills that they can get around fine, whether it's with a dog or a cane or whatever it is. But he's been trying, and he has been successful training people to actually use this kind of echolocation. The thing is, when a person first starts out trying to do this, it's difficult. There's kind of a little bit of a steep learning curve, as you can imagine. But even after the first day, you can go online and look at another video. You can see how they actually acquire the ability to do this. And he also takes kind of a hands-off approach, so he won't let them walk into the middle of the street and you know, get crashed. But if they make mistakes, he doesn't really care. He wants them to be resilient when it comes to being able to use this and not being reliant on other things around him, like a seeing-eye dog or another person or whatever. So there's actually this big controversy going on between a lot of these organizations for helping the blind and him. Because a lot of them, this is how they've always done things. They've made things so that blind people have all these things they can kind of use to help them get around in the world. Whereas he's trying to make them actually more self-sufficient. So, just so you get the idea. Okay, now, so I just wanted to kind of give a more in-depth detail of what's actually going on with the moths and the bats. To actually look at the different levels of proximate causes, including the signals, how they're making the signals, the nervous system that's developed around that, and everything for both sides of the equation. It turns out that we talked about voles before with their different mating behaviors. And if you look at the bottom there, that's kind of prime vole habitat. If you actually got down on your knees and stuff, you'd see these different runways that the voles build and use to travel across that. So there's a kestrel, which is a very small falcon. It's actually a European kestrel, but we have one just like it here. And it turns out that kestrels can actually see UV. And you think, well, whoop-de-doo, that's great. Turns out a lot of birds can see UV, and a lot of insects too, in terms of pollination syndromes and whatnot. But the question has always been, how do these birds know whether there's a lot of voles there or only a few? And it turns out that the voles, unfortunately, leave a lot of droppings and a lot of urine, and those actually show up under UV light. Okay, they fluoresce. And since the kestrel can actually see that, they can fly over a field and say, well, this is good hunting. And versus, well, you know, the grass is always greener on the other side of the hill. Okay? So, and then what they actually did with this, and this is, obviously, they put it in terms of hypothesis. So the question is, how do these kestrels figure out whether there's a lot of food or not? And I don't know why they thought of this, but they thought of this hypothesis. And they said, okay, if that's really true, then we actually have to show, first of all, that urine and feces from these voles fluoresce, and then actually taking a kestrel into a laboratory and seeing how it responds to signs of voles or not voles. And that's exactly what they did. Okay? So this is, you know, it turns out that a lot of animals are better at, or they can do more with what they have. They see in the UV spectrum, which we can't. They also have other cones that, and instead of just blue, red, and yellow, they have other cones as well for different colors as well. And just like echolocation, we can't hear above 20 kilohertz, but a lot of other animals can. Oh, and by the way, the voles, I've been in fields where the voles, there were like 1,000 per square acre. Okay, I mean, it's literally teeming with voles. Okay, and then in terms of neurology and migration, there's a bunch of different things that a lot of animals use in terms of migration and figuring out where they're going, and in fact when they should leave and when they should then head back the other direction. Okay, so there's two major migration routes. So those west of the Rockies, they head down along the coast here. You can go up to Oxnard just about this time of year, and you can see eucalyptus trees filled with monarch butterflies. Okay, now it turns out the main migration route, or the ones east of the Rockies, they all funnel through and actually go down into central Mexico, which is the ones you've probably heard about. In terms of, there's this area, pretty small area, kind of old growth forests, and literally millions and millions and millions of monarchs winter there. They hang in the trees, kind of like curtains. Okay, and also the monarchs have they actually multi-generational migration. So when they actually go from the spring migration routes, when they actually go back up, let's say up to here, they actually land in Texas and New Mexico, and they lay their eggs. And they lay their eggs on milkweed, which is a toxic plant. It has cardioglycosides as well. And then monarchs then incorporate their larvae, caterpillars incorporate the cardioglycoside in a tissue. They mature, and then they continue on their journey farther north, where they finally start breeding up along into Canada, the northern U.S., along the Great Lakes, all that. Okay, so, and then finally, when it's fall, they turn around and they actually fly all the way back in winter, and then the cycle begins again. They're not doing so well now, just so you know, but you can actually go see them. They're kind of a protected area, but there's been, well, it hasn't been very good. Okay, now, monarchs, it turns out, can use UV radiation to begin their journey. So, and they're able also to, so they use that in terms of UV, well, any radiation changes depending on what latitude you're at. So they can read that. Now, then they also switch to polarized light in terms of when they actually use orientation. And I think it's, let's see, let me look at this exactly. Okay. So, okay. Right, so what they did was they actually put filters on the lights that they had in a lab, and they showed that, in fact, they used polarized light, especially in the early AM or late sunset, to figure out exactly kind of where they are. So it just shows that butterflies with a horizontal filter oriented at 90 degrees north-northwest markings with no filter or vertical filter oriented towards the south-southwest. Okay, and actually you can actually see this. Okay, so south-southwest, and then north-northwest for those that had the filter on. Now, there's other things that, so they've got photoreceptors that they can use that, obviously. There's other animals that I'll probably call it a day, but pigeons, they actually use magnetic information. So we'll get more into that when we get back. We'll have the exam on Wednesday of next week.
There's no school on Wednesday.
What?
Wednesday's a holiday.
Wednesday's a holiday?
Veterans.
That's not good. How can we do that? Okay.
Take-home exam?
What?
Take-home exam?
You want to do a take-home
Yeah,
exam?
give it to us on Monday and then we'll bring it back on the next one. Bam.
I'll think about it.
Problem solved.
I'll let you know soon. But then, of course, I have to make it a lot harder.
There you go.
Mm. I do. So, I'll think about it, and I'll let you know soon. Veterans Day? God, that's crummy.